Okay, so adaptive immunity is, we're considering it the third line of defense. Okay, so it's when your body actually starts responding to specific pathogens or specific antigens. Okay, now an antigen is anything that causes an immune response. So anything that causes an immune response. So it can be cat fur, it could be a bacterium. Okay, now we're going to talk about lymphocytes. Okay, so 3 types of lymphocytes circulating in your blood. 80% of them are T cells, and they're responsible for cell-mediated immunity. 15% are B cells, which is they're going to make antibodies. And then 5% are natural killer cells, which are nonspecific. They're going to kill, destroy anything they recognize as non-self. Okay, so I just kind of jumped the gun and told you this. So an antigen is a substance that binds to a component of the adaptive immune system and causes an immune response. It can be a protein, it can be a glycoprotein, it could be a lipoprotein, it could be a polysaccharide. Okay, so it's some component of, it could be cat dander, okay, it could be pollen, okay, it could be a virus, it could be a prion, which is something like that's an infectious protein. I mean, it could be bacteria, fungi. I mean, people are allergic to anything, okay? So it creates an adaptive immune response. Okay, now we mentioned this before, so interleukins, we always abbreviate them IL. Okay, so interleukins are, they're saying hormone-like messengers between white blood cells. Okay, so this is how white blood cells communicate. So it's the cytokines or the communication between leukocytes, between white blood cells. It's how they're talking. Okay, so there are lymphokines, which are going to be produced by lymphocytes, and monokines, which are produced by macrophages. So that kind of makes sense. Okay, now here's the types of T lymphocytes. So first kind are helper T. Now the type of receptor that's on them that's specific for the helper Ts are called CD4 receptors. So these are like the general of the army. Okay, so these are going to be in control. Okay, so CD4, sometimes they call them T4s. So these are the ones that are in charge of adaptive immunity. Okay, and this is the type of receptor on them. Cytotoxic T cells have CD8 receptors. Okay, so these are going to be more, these are more like the infantry. So they're going to be able to kill things. Okay, now the suppressor T cells, they're going to turn down the immune response. Okay, so they're going to suppress it. We don't want it to just keep going on and on. We want to be able to turn it down. Okay, and then memory T cells are going to remember what you've been exposed to. Oops. Okay, so if you've been exposed to chickenpox, okay, you create memory T cells to the chickenpox, and then the next time you're exposed to chickenpox, you will create an immune response to it faster because you have memory. Okay, so memory they keep track of what you've already seen so that you can respond to it faster. Okay, now we want to talk about the types of immunity. So natural immunity is something that you acquire, okay, and artificial is something that we give to you. Active means that you are developing the antibodies to it. So natural active would be let's say you get the chickenpox. Okay, so like if you're my age, we got the chickenpox and we developed the antibodies to it. Okay, so we developed the antibodies to it, and it was easy for us to fight it off or the next time we got exposed to it. Okay, so natural exposure, okay, and active, your body developed the antibodies. Okay, now artificial active would be like a vaccine. Okay, so now kids get vaccine for chickenpox. Okay, so we shoot a little bit of the chickenpox antibody or antigen into them, and they develop active immunity. They develop the antibodies to it. Okay, natural passive would be mom to child. Okay, so it would be through the placenta or through breast milk. That's how that one works. That doesn't last forever, right? You only keep it as long as, 6 months, 4 months. Okay, then artificial passive would be like if you have a snake bite or a spider bite, and we give you anti-venom. So we're giving you the antibodies to it. So that's not going to last very long at all, okay? So we're giving you the antibodies for it. Okay, now, how does the immune system recognize itself? So we have these things that are called MHC markers. So MHC stands for Major Histocompatibility Complex. Okay, so basically we have these MHC markers on our cells, and everyone has their own unless they have a identical twin, and then they might have someone that has similar markers or has the same markers. Okay, so we have 2 categories of MHC markers. So one is they're found on all nucleated cells. So every cell that has a nucleus has these MHC1 markers, and that's how we know, our body knows that those are our cells and that it shouldn't attack it. Okay, MHC2 markers are on antigen presenting cells. So those are called APCs. So antigen presenting cells actually will present a piece of the antigen so that the rest of the body knows that you killed it. Okay, so if you have, say, a white blood cell that comes in and it kills strep. Okay, so let's make it green. Okay, so it comes in, it engulfs, whoops, I wanted this to be like this to be like this. It engulfs strep. Okay, after it engulfs it, it will place a piece of the strep organism on the surface of it at these MHC2 markers, and it'll say, look, I killed strep. This is what I did. Okay, so it presents a piece of the antigen so all the other cells know that it killed that and that they should be aware and looking for this green organism. Okay, so here's a picture of kind of how it works. Okay, so we phagocytize the antigen. Okay, so there's the green organism. Okay, here's the lysosome. The lysosome fuses with the vesicle that's got the organism, the pathogen in it. Okay, it destroys it. Okay, it breaks down the antigen, breaks down pathogen. Okay, we get rid of the waste, and then we also display some of the pathogen on the surface. Okay, and we're displaying it at these MHC2 things, and we're saying, hey, look, we killed it. So B cells and macrophages are antigen-presenting cells. So they let everybody else know what they've seen. It's kind of like a warning system. It's sending out an SOS. This is what we killed. Or you could think of them as show-offs. Oh, this is what we killed. Okay, now, helper T cells, what they're going to do is I always think of them as a general. Okay, so they're going to coordinate your adaptive immunity. Okay, so they're going to coordinate humoral and cellular immunity. Okay, so here's the cell that's antigen presenting. Okay, so it's saying, I killed strep. Okay, so it binds to the helper T cell, and the helper T cell is going to trigger nonspecific defense. Okay, it's going to trigger the production of antibodies, and it'll trigger these cytotoxic T cells to go around and kill. I'm not worried that you know all of this. Don't worry about all that. Just kind of know how this process works in general. Okay, now T4s, they have that CD4 receptor. Okay, HIV, when it infects these helper T cells. Okay, so if you take out the general or the army, okay, then you have a weakened immune system. Okay, so cytotoxic T cells, they're the only T cells that directly attack enemy cells. They have what's called a lethal hit mechanism. So they release perforin, so they put a hole in the cell wall or the cytoplasm. Okay, we have lymphotoxins, which can destroy target cell DNA, and we have tumor necrosis factor, which can kill tumor cells. Okay, now suppressor T cells, and I mentioned this before, as the pathogen numbers go down, the suppressor T cells slow down their immune response. Okay, so they are there to help prevent autoimmune disease. Okay, so they're supposed to help prevent us from having an overactive immune system where our immune system starts attacking things that it shouldn't. Okay, memory T cells, they're going to remember, okay, so that we can recall quickly and mount a faster secondary response. Okay, so the first time you're exposed to something, it's a primary response. The second time you're exposed to something, it's a secondary response. Okay, now, humoral immunity. Okay, so you're getting like 3 slides with humoral immunity. Okay, so what happens for humoral immunity, what I really want you to know is that these are antibodies. Okay, the B cells, so B lymphocytes bind the antigen, they take it in, digest it, and they're going to present it. Okay, they get stimulated by the helper T cell, okay, and they divide, and then they become plasma cells. The plasma cells are what actually produce the antibodies. Okay, now the antibodies are Y-shaped proteins, okay, so they kind of look like this. And what they're going to do is they're going to bind to the antigen. They could take it out of circulation so it's not harmless anymore, or they can tag it so other cells could destroy it. Okay, we also have memory B cells, and we keep those so that we can make antibodies faster. Okay, now how do antibodies work? Okay, so humoral immunity, remember that's what's antibodies. Okay, so we can neutralize. So we could, if this is an organism, the antibody can bind to it, and then this can't cause any infection anymore. Okay, it could trigger complement fixation. Okay, so the complement was that set of proteins coming from the liver. Okay, and then it could cause agglutination. So it could cause, and I have that on the next picture, and it couldn't cause precipitation. So yeah. So agglutination, so here's antibodies bound to red blood cells. Okay, so agglutination, what happens is it's going to cause the red blood cells to clump. Okay, and if they all clump together, they're not functional anymore. Okay, and then if we precipitate, what happens is we take it out of solution. And then really a macrophage could come up and engulf this and get rid of it a lot easier than it could if it was just, the antigen was just floating around out there by itself.